cistern model. So Golgi apparatuses actually look like they're undulating in space. Yeah, Adam. Yeah, for sure. Do you want me in the picture or out of the picture?
What do you mean by correct? Like, your body just does it, doesn't it? Like, or like, what does it, like, I don't know. What do you mean by correct?
What do I mean by correct?
Yeah.
What I mean is, what does the cell actually do, and what does the Golgi apparatus actually do? And it actually does the cisternal maturation model, not the static cistern model. So the reason I gave you both of those hypotheses, or both of those models, is because there was a big debate about how it was the Golgi was actually processing things. So competing research groups were looking at both of those models, and what we've come to find is this one is more correct than this one is. But like I said, sorry, like I said, it's probably some combination of the two, leaning towards this one. Yeah.
That was my question. Does it sometimes do the static?
Yeah, it's probably more like a continuum. I mean, probably, and this is, okay, so this, we're gonna leave like the safe harbor of fact, and we're gonna talk about my speculation now for a few minutes. But what I think probably happens is, so proteins will remain within a cistern for a while, and then that cistern will move, and then after it's moved some number of steps, the protein will move to a next series of cisterns that will then move along. It's probably what happens.
How long does it usually take for the protein to, like, how long does it stay in one cistern for?
That is a wonderful question, and I don't have a great answer for it. And the primary reason for that is the kinds of proteins that we make are incredibly varied. And so if it's a simple protein, it won't stay in the cisterns very long, right? Because the ribosomes can make most of that protein before it actually gets to the Golgi apparatus. If it's a really complex protein, like if a single protein has multiple protein subunits associated with it, then it's going to remain in the cisterns much, much longer. But I think an approximation would be like portions of seconds. It happens very, very quickly. Yeah, it's not like hours or days, for sure. It happens really, really quickly. Good question. Others? Okay. So y'all are okay with this idea of the Golgi apparatus so far? Okay. So we're going to move on to some more members of the endomembrane system. Now, the endomembrane system so far consists of our endoplasmic reticulum, the vesicles, and the Golgi apparatus. And it's really difficult to call vesicles organelles by themselves because they're really just transport bubbles. This next organelle, however, the lysosome, is a really interesting one. And I think it's interesting because if you look at the cell really is analogous to our entire body. All the things that our body does, the cell is capable of doing on a much finer scale. So you could really think about the nucleus as a brain, right? I mean, it's kind of directing sorts of things. Another function that our body accomplishes is digestion, and that's the job of the lysosome. So there are intracellular stomachs, basically, and that's how you should think about the lysosome, as an intracellular stomach. It's something that collects organic material, breaks it down into constituent parts, and then redistributes that within the cell. Okay, so our lysosome. Does anybody know the band Fish? Spelled with a PH instead of an F. No. Well, you guys should look up Fish. It's a jam band from like the 90s, and they have a song about the Golgi apparatus and the lysosome. So every time I teach this content, that song starts going through my head. Anyways, the structure of the lysosome, it's a smooth membrane sac. It's just like a bubble of membrane, and it's just a single layer of membrane. But remember, that membrane consists of it consists of a phospholipid bilayer, right? So there are two layers of phospholipids. But it's not just a sac. The sac has embedded on the inside of it hydrolytic digestive enzymes. So we're going to say studded with hydrolytic enzymes. And a hydrolytic enzyme is one that breaks stuff apart. So it goes through the process of hydrolysis. And remember, hydrolysis is the opposite of dehydration synthesis. So it's taking water and it's cleaving a covalent bond apart. Okay? So this is a digestive system. Its function is to digest organic molecules. So things like an amoeba or a paramecium that are unicellular, this is what they have for a stomach. They're able to engulf things, bring them into their bodies, send them to the lysosomes, and the lysosomes break all of that organic gooey stuff down. We also think of it as like a recycling center, because sometimes cellular components, whether they be whole organelles or whether they be molecules, are malfunctioning. And the lysosome is able to take those, break them down into constituent parts, and then reabsorb them so that our cells can make new, better stuff out of them. Sometimes, like organelles go wrong, right, like a mitochondrion, and if it's repairable, then our cell will repair it. Sometimes, however, that repair is beyond, or excuse me, that organelle or the cell is beyond repair, in which case the lysosomes become a part of a huge cascade of events in which a whole cell is completely digested and broken down. Can you think of a context in your body where you'd want to just kill a whole cell? Bacterial infection, sure. Maybe if it's so infested with some bacterium that you just kill off the whole thing. I like it. Can you think of others?
Aren't there what is it? Like the white cells and like the red cells, some of them like attack each other? I forget.
So white blood cells
Yeah.
are a part of our immune response, so they're able to identify problems and take care of them. Yep, for sure. Yeah. Cancer is a wonderful one. So cancer is interesting. We're going to talk more about cancer when we talk about mitosis. But what happens is cells begin to divide beyond our body's control. We're no longer able to say stop dividing, right? But and all of us at some point in our lives has had a cancerous cell. Generally speaking, our bodies are really good at identifying those and taking care of them. And the lysosomes are an important part of taking care of problematic cells. Sometimes, though, those cells slip past our ability to identify them or control them. And then they begin to grow and divide beyond our body's control, causing tumors. And that's what a tumor is. It's just a growth of cells that our bodies can't control. Okay, another closely related organelle is something called a peroxisome. We've actually already looked at peroxisomes in laboratory, though you didn't know that we were looking at peroxisomes. Peroxisomes have a similar construction to lysosomes. They're just a membrane sac that has digestive enzymes in it that's able to break down, but specifically detoxify compounds. So that's its job, it's detoxing the cell. Kind of like the smooth endoplasmic reticulum did as well. Oops. The structure of the peroxisome is a smooth membrane sac with digestive enzymes. And the function is detoxification. So peroxisome sounds a lot like hydrogen peroxide, right? And we looked at liver and hydrogen peroxide last time in laboratory. So the peroxisomes have that enzyme called catalase that's able to take hydrogen peroxide and split it into water and oxygen gas.
What does that say? With what? Enzyme?
Yep. Yeah, I know, I shouldn't even try and write on the board because it's illegible. And then the function is to detox. Okay, so there's a disease that I'm going to want you guys to be familiar with, associated, sorry, was there something up here that wasn't legible? This says smooth membrane sac with enzymes.
Okay.
Yeah. The screen got in the way. And then the function is to detoxify stuff. So in plants, when fats and sugars and proteins and stuff build up and it becomes toxic, the peroxisome sucks those up and breaks them down. And it's cool, I mean, if you looked inside of a peroxisome, the enzymes have like this really highly organized structure. It's almost like a crystalline structure, and it looks like a big square like this. And that's the business part of the peroxisome. Okay, so there is a disease that I'm going to want you to be familiar with, and it's called Tay-Sachs disease, and it's a disease associated with the lysosome.
How do you spell that?
Tay-Sachs, Tay-Y-S-A-C-H-S. So this is an important one that we're going to come back to, much like sickle cell anemia throughout the semester, because it illustrates a number of really important phenomena. The first of which is, and most important for our discussion today, the bad stuff that can happen when organelles go amiss. So one of the primary jobs of the lysosome is to identify concentrations of stuff that's too high and to break it down. So if we start building up too much protein within a cell, lysosomes can grab those proteins, break them down, and redistribute stuff. If we get too much fat inside a cell, we can do the same thing. And all of our cells should have lysosomes. One of the more important groups of cells in our bodies are our neurons, the brain cells. Tay-Sachs disease is a disease associated with lysosomes in the neurons. In individuals with Tay-Sachs disease, their lysosomes are able to identify fats inside the cell, and so it consumes those fats, it pulls them into the lysosomes, but they lack the digestive enzymes to break down those fats. And so the lysosomes become so engorged that they actually destroy the neurons from the inside out. And when that, basically it causes the cell to lose its shape. And when it loses its shape, it loses its functionality. So it's a genetic disorder really associated with an inability to produce the right kinds of digestive enzymes for the lysosomes. Infants with Tay-Sachs disease typically begin exhibiting these sorts of characteristics at like 4 to 6 months, really, really young. And it's usually lethal within the first year. There is no treatment, it's a genetic disorder. What's interesting about it is that there is a very high occurrence of Tay-Sachs disease among a group of the Jewish nation called the Ashkenazi Jews. The Ashkenazi Jews live in northern Europe, and they left Jerusalem hundreds of years ago. And when they left Jerusalem, they took with them, and all societies, all ethnicities have some background amount of Tay-Sachs disease that just naturally occurs. It's a genetic disorder that's just kind of ubiquitous across all of our genomes. But when the Ashkenazi Jews left Jerusalem, they took with them, as a product of random chance events, a higher proportion of the genes for Tay-Sachs disease than the rest of that Jewish nation had. And because of religious constraints, there was a high degree of inbreeding within that society, right? Because individuals of the same ethnicity and individuals of the same religious persuasions like to have families together. So as a product of inbreeding, that Tay-Sachs disorder became fixed within the population. And so now, among the Ashkenazi Jews, Tay-Sachs disease is very abundant. It's fixed within that population. Their children contend with it much more than any other ethnicity does. So I tell you that second part of it because what that illustrates is a mechanism of evolutionary change. Remember, evolution is just a change in gene frequencies. And when that portion of the Jewish nation emigrated, it took with it a relatively disproportionate sample of that one gene. Consequently, its genetic composition is quite different than the composition of the rest of the Jewish nation. And that's evolution. Okay, so this shows you the functionality of a lysosome, and it does it in two ways. That is, it illustrates two processes, the first of which is called autophagy, or autophagy, or autophagy, or self-eating. What's happened, right, is this mitochondrion that we haven't talked about yet is malfunctioning. It's not working the way that it should. And so Mr. Lysosome comes along, and it binds to that mitochondrion, and then it completely digests it, and then distributes the parts of the mitochondrion back out into the cell. So the cell can make new and cooler stuff from those things. There's also a process called phagocytosis. And so phago refers to eating, and cytosis refers to the cell. So cellular eating. There are some organisms that consist of just a single cell. And what's happened here is this cell has recognized this thing as something yummy that it wants to eat. And so it pulls it inside the cell, and then the lysosome again comes over, binds to it, digests it, and then distributes all of those yummy things into the rest of the cell. Okay? Questions about the process of digestion by the lysosome? Yeah, Carly.
So autophagy is when it happens already within the cell, and
Phagocytosis?
the second one is when it's something